This is USMLE QA, USMLE material presented in a question and answer format structured similar to the first aid for the USMLE Step 1 book.
Question: what is trimming? Answer: trimming is the removal of N or C terminal propeptides from zymogen to generate mature protein. Question: what are covalent alterations? Answer: covalent alterations are modifications to proteins that involve the formation of a covalent bond between the protein and another molecule. Examples include phosphorylation, glycosylation, hydroxylation, methylation, acetylation, and ubiquitination. Question: can you give an example of a protein modification that involves phosphorylation? Answer: one example of a protein modification that involves phosphorylation is the activation of enzymes by protein kinases. Phosphorylation of a specific amino acid residue on the enzyme can change its activity. Question: how does glycosylation modify a protein? Answer: glycosylation is the process by which a carbohydrate, such as a sugar, is covalently attached to a protein. This modification can occur on the extracellular side of the protein and can affect the protein's stability, solubility, and activity. Question: can you explain what methylation is as a protein modification? Answer: methylation is a type of posttranslational modification in which a methyl group is added to a specific residue of a protein. This can have a wide range of effects on protein function, including the regulation of protein-protein interactions, DNA binding, and transcriptional regulation. Question: can you explain what ubiquitination is as a protein modification? Answer: ubiquitination is a post-translational modification in which a ubiquitin protein is covalently attached to a lysine residue of the target protein. This process is catalyzed by E3 ubiquitin ligases. This modification can lead to protein degradation by the 26S proteasome, or can also target proteins for other cellular processes such as endocytosis or DNA repair. Question: how does hydroxylation modify a protein? Answer: hydroxylation is a post-translational modification in which an oxygen atom is added to a specific residue of a protein, typically proline or lysine, via the action of enzymes known as prolyl hydroxylases or lysyl hydroxylases. This modification can affect the protein's stability, solubility, and activity. Question: can you explain what acetylation is as a protein modification? Answer: acetylation is a post-translational modification in which an acetyl group is added to a specific residue of a protein, typically lysine. This modification can affect the protein's stability, solubility, and activity by altering its charge and changing the way it interacts with other molecules in the cell. Question: can you explain the role of post-translational modifications in protein function? Answer: post-translational modifications play a crucial role in protein function. They can affect the stability, solubility, and activity of a protein, as well as its interactions with other molecules in the cell. These modifications can also help regulate the activity of enzymes and other proteins, and can be used by cells to respond to different signals and environmental changes.
Thank you for watching our Q&A format video. We hope this helped you learn, review, and retain knowledge needed for the USMLEs. For more information on this topic, check out the link in the description. Now, if you want to take your USMLE prep to the next level, be sure to check out all our courses, which are structured to follow along with the first aid for the USMLE Step 1 book. You can find us at usmleqa.com. And if you're watching this on YouTube, be sure to like and subscribe.